Recombinant human growth hormone. Can there actually be a natural alternative? Now keep in mind this is an animal model, so we have to see if it translates over to humans. But let's look at this graphic real fast. We have G0, that is your control group, they're not doing anything. You have G1, G2, and G3. G1 and G3 is 304 milligrams of this Keyfobell powder, the researchers deemed it, and that is Astragalus, membranaceus, red ginseng, and young deer antler, either from components from either the red deer or the sika deer. And then G2, which is just a higher dosage. So if you look at the G2 and P, let's not forget P, believe it or not, is recombinant human growth hormone injected. So when you look at the G2 to the injection of growth hormone, it's pretty darn comparable. Now, also too, in some of the charts here, I put a red line through. Those are ones to highlight the most dramatic differences. Even in the low dose of the Keyfobell powder, Astragalus, membranaceous, red ginseng, and components of young deer antler, you still had phenomenal results compared to the control. Now, a little bit of the backstory here is as follows. The researchers were trying to find safer alternatives for children which are growth hormone deficient and basically facing growth issues, especially in regard to stature. So we're looking for something safer. And if this animal model does translate to humans, they may have done that. So without further ado, let's get right into the research as follows. The effects of novel astragalus-based extract Keyfobell powder, otherwise deemed KFB, on longitudinal bone growth via insulin growth factor 1 upregulation, a potential growth hormone alternative. Now keep in mind, this has to translate to humans, and this is an experimental animal model. Backstory. Studies have demonstrated that ghrelin potentially stimulates growth hormone secretion in humans. Growth hormone deficiencies occurs when the hypothalamic or pituitary disorders disrupt growth hormone secretion. Please forgive me if I speak a little fast, a lot of territory to cover. A decline in growth hormone secretion with age is associated with decrease in growth hormone releasing hormone, GHRH, just something to keep in the back of your head. Therefore, growth hormone is widely used for the treatment of growth hormone deficiency or short stature. However, its therapeutic use can lead to adverse effects, including cell proliferation, which may increase the risk of cancer. In addition, growth hormone therapy may induce other complications such as diabetes, fluid retention, joint and muscle pain, hypertension. Owing to these effects, the application of growth hormone remains limited, particularly in pediatric populations. Hence the research. In Korea, traditional herbal medicines are used to treat growth challenges in children, in particular, astragalus extract composed of the stem of Deuterococcus centicosus, interesting, and the roots of Astragalus membranaceus and Flumbus umbrosa, which have been evaluated as promising therapeutic candidates, as it may mimic the effects of growth hormone without the associated risks using rats as well as humans. The presented study investigated the effects of novel astragalus extract, Keyfobell powder, composed of the roots of Astragalus membranaceus, red ginseng, and of course young horn of either the red deer or the sika deer on bone growth and development. These effects were compared with recombinant human growth hormone somatropin as a positive control. Bring back to the graph we had earlier, which is synthetically produced form of human growth hormone that functions by replacing growth hormones in the body. It is used to promote weight gain and growth in pediatric patients with specific conditions that inhibit normal growth and development. Hence the search for something a little safer. The results. In our study, quoting, the Keyfobell administration resulted in elevated insulin growth factor 1 comparable to those achieved by recombinant human growth hormone, with even higher levels of insulin-like growth factor binding protein 3 in the Keyfobell group compared to the control group. The increased insulin growth factor 1 and insulin-like growth factor binding protein 3 levels suggested that the Keyfobell to induce a growth hormone-like effect without adverse outcomes associated with growth hormone therapy. In addition, Keyfobell enhanced the expression of bone morphinogenic protein 2, something to think about as far as maintaining bone strength, in bone tissues. BMPs are members of the transforming growth factor beta superfamily that are essential for both embryonic development and adult bone maintenance. Keep that in mind. also essential for adult bone maintenance. These proteins play crucial roles in bone remodeling and continuous and essential process within the mature skeletal system. The observed increases in insulin growth factor 1, in the insulin-like growth factor binding protein 3, and the bone morphinogenic protein 2 levels after Keyfobell administration demonstrated its multifaceted effect on bone growth and suggested it as a promising natural alternative to conventional growth hormone therapy. Now here's the methods. You can see the ratios, the sources of each component of the ingredient. Again, Keyfobell is not like an item itself. It's something that the researchers basically just christened it as follows, as far as the Astragalus, red ginseng, and the components of young, antler from either red or sika deer. So basically, there you have it there, including the ratios as well as far as the preparation. Now, as follows, too, to show you using Gemini Advanced to give you an idea of the comparison that may play a role. It's far more complex than just basically bone body surface area per se. Conversions from basically mice to humans. But it gives you an idea. It may not be an extravagant, large amount per se that's required to maintain or generate the same outcomes in humans as we have in animals here. Again, that's just an example. You can see the different ones that we have, the 3 to 4 milligrams, either in saline or milk. I used the low dose as the comparison, and of course you have a conservative safety margin as follows. Conclusion, again, just an example. Conclusion, Keyfobell powder increases the transcription of growth hormone releasing hormone and ghrelin in hypothalamic cells. Keyfobell administration resulted in a notable dose-dependent increase in body weight and bone growth. In vivo, the observed changes in insulin growth factor 1 and insulin-like growth factor binding protein 3 levels were comparable to those observed in the growth hormone-treated group. And remember, the Keyfobell powder is being done orally, and basically the recombinant human growth hormone group is done through injection. These findings indicate that the Keyfobell may function as a growth hormone mimetic substance that stimulates bone development by increasing insulin growth factor 1. Consequently, Keyfobell has the potential to support children healthfully with idiopathic short stature or growth hormone deficiency. That is just groundbreaking and quite amazing. Again, gratitude to the researchers. That's phenomenal. The application has such a wide use to help so many people. I am humbled you watch and look forward to see what you and I share next week. All right, catch you all next time. See you then. Bye.